once again po, a very good morning to everyone, and thank you for joining us again at the second day or part 2 of our webinar series entitled Livestock Biotechnology Center Research, Development, and Extension Agenda Technical Caucus webinar series. So once again, my name is Janlo P. Baligad, and I am the moderator for today's webinar session. On behalf of Livestock Biotechnology Center and BCC, I would like to extend a very warm welcome to all of you. We appreciate you taking the time of your busy schedules to join us today. We hope you will find the program we have lined up for you to be fruitful and engaging. We are indeed honored to have you here with us. We have about 55 participants yesterday, and now, as of now, we have about 35 participants from different government agencies and academic institutions around the country. As I am said yesterday, our webinar series is initiated by the Livestock Biotechnology Center together with the Philippine Carabao Center. This online activity aims to disseminate innovative livestock biotechnology research results with appropriate policies and advocacies for a competitive, profitable, and sustainable livestock industry even amidst the pandemic. Yesterday, we talked about RDE agenda and advocacies of the DA Biotechnology Program Office and the role of biotechnology to the society and its current implications with Ms. Joy Humalon, the Project Management Officer of DABPO as the presenter, and a presentation on genetic diversity delivered by Ma'am Lillian Villamor of Cryobank Unit, PCC, National Headquarters, and Chinpool. And now, we are in the second part of this webinar series, and this is composed of two presentations focusing on breeding selection and repro biotechnology. So, before we begin, I'd like to share a few reminders. First, please be informed that this webinar is being recorded, and while speakers are in session, you may send your comments and questions to the chat box. Please remember to indicate to whom you're addressing the question to. Also, in asking, you may introduce yourself. And at the end of the webinar series, we will be sending a post-webinar assessment form via email. And after you send us back the post-webinar assessment form, we will be sending a certificate of participation to all the attendees. And napakaswerte po natin ngayong araw na ito dahil ang mga magiging speaker po natin ay talaga naman pong eka nga po ay prolific and productive researcher dito sa ating bansa at higit lalo po dito sa Philippine Carabao Center. Ngayon, hindi na po natin papatagalin. Okay, so let us proceed to our first presentation this morning. And our first speaker is a graduate of Doctor of Veterinary Medicine at the University of the Philippines, Diliman, Quezon City. She took her master's degree in animal science at the University of Arkansas, USA, and her PhD in agriculture. And ladies and gentlemen, please join me in welcoming Dr. Esther B. Flores. Hello po, Doc.
Hi, thank you for the nice introduction.
Okay, Doc.
Okay, hopefully we will have a very fruitful and informative session today. Welcome sa mga participants natin. Thank you for giving me the opportunity to present some of the outputs of our research in our laboratories. So I'm not going to take a lot of salita pa, but I'm going to share you my presentation. I'm excited actually to share what our recent effort is about. So here it is. I'll just fix the, okay, so I'll just fix the, what do you call that? And the itsura. Okay, so I'm going to talk about the genetic improvement program and molecular genetics in a sense that I'd like actually to see, to show you where the molecular genetics comes into the conventional breeding program implementation that we are doing in PCC. Okay, so essentially, when we talk of breeding program, most of the time when they say, I used an imported breed and cross it to my native, they think that that's actually already the genetic improvement. But in fact, that's actually just a crossing. When we say we have a breeding program, there should be several components. There's actually a structure to it, and such that whatever effort you put into it is actually going to be a cycle. So you will see dito, nakaikot na to, no? So it's a never-ending cycle. And unlike in other fields, actually, when you talk of breeding, it's actually really a never-ending cycle because you have to keep on pushing. You have to push the selection intensity forward so that the whole population is changed because you're working on shifting the population average from one generation to the next. So you build upon the gain from the previous generation and work on it today. And then you plan the next generation again by selective breeding and identifying the best animals that are actually going to be used for breeding. And at these various components of yung cycle natin, the application of molecular genetics can have a role, di ba? But there are some aspects here that mas importante talaga yung molecular genetics kesa sa ibang aspect. Kunwari, where do we see the application of molecular genetics or DNA marker technology? For one, in the collection of information, when we talk about family relationships, we want them to be as accurate as possible. So may application tayo dyan. When we do the genetic model, we can actually incorporate it. And then when we select individual animals on their basis of their genetic merit, may application ang molecular genetics dyan. So I'll go further in a sense that where do we actually apply it? We know that when we say, hindi ko naintindihan yan. Well, there are some technology applications that is not suitable for, let's say, sa commercial. Most of the time, we apply yung ating marker, DNA marker technology sa mga nucleus herds, the ones that are actually going to carry and disseminate the good genetics to the rest of the industry. Bakit? Ang lalaki, or the bull, or the boar, the buck, or anyway, it's the male actually wherein sa kanya fitted or suitable lahat halos technologies, except for some. But most of the time siya, the male of the species is actually the ones that actually get the full benefit of the application of molecular genetics in animal breeding because they are the ones that actually carry or disseminate the genetics to the rest of the population. And you will notice my generation lag, but it is actually here, this upper small triangle of a pyramid structure ng animal breeding where our technologies is actually applied to the fullest. Okay? So, very first, sabi ko kanina, sa cycle natin, the very first one that actually I said is the why. For the conventional genetic evaluation, we actually rely on, we call it pedigree-based blood evaluation or genetic evaluation. So when we say pedigree, you're talking of the traceability, the nanay, the tatay, probably the grandsire, the granddam, the grand-grandsire. So that's the pedigree or the family tree. And the more that is accurate, the better is your evaluation. Yan. Okay. So in terms of traceability, napaka-importante ng tamang pedigree sa genetic improvement program and evaluation, and also in the breed registry of some of the animal associations. Parang libro yan ng mga genealogy. Okay, so other uses for traceability are for disease control, surveillance, but for our topic today is actually we are looking at how important the traceability is in the accuracy of identifying the individual animals that carry actually the very good genetics. sinabing tama ang tatay and it's really good, then they get the full benefit, the industry benefits from the good genetics is actually going to be disseminated to the whole population. But kung mali naman yan, you actually hindi mag-benefit, baka nga makasama pa. So very important ang registration. That's why for most countries actually, they incorporate what we call parentage verification bago nila i-issue ang pedigree certificate for the purebred bulls or animals that are actually breeders, future breeders of the next generation. See, this is very important, parentage verification. And how do you do this? Sabi na, magaling kami mag-record, but there are inherent errors sometimes. Tao lang tayo. There are errors in the recording kung yung dati pa. Now, here comes the new technology. We actually verified it, a pedigree by, for one sa PCC, what we have right now is a microsatellite-based parentage testing. So we know that microsatellite is a DNA marker. It's an unfolding, but it is very, very useful for matching the size of the alleles of the specific microsatellites of the parents, the tatay, and the dads. And you can have like, hindi ka sure kung siya ang tatay, baka kakwit, dalawa sila, tatlo sila, I'm not so sure. We can actually run through several putative parents or potential parents and match this against the calf. And we can verify that. So pag minatch, ay, hindi yan ang tatay. So we have actually what we call a 99% confidence that we can, in the system, we can actually take out the non-parent, yung more likely non-parent. This is a typical example. So, mapapansin niyo on this side is what we call the load score. This actually gives you confidence. So, the higher the score, the more confident you are na tama yung pagkaka-identify na reported tatay at saka reported nanay or the reported sire and dam of a particular calf. We're talking here of individual animals. So, you will notice negative. So this is match against the candidate father. Usually the dam is not a problem. It's always the sire. I don't know why. Baka sa tao ganun din. It's always the sire. Sometimes that is normally reported wrong. And you will notice here there are actually 3 markers out of the 14 compared between the calf and the sire. 3 are mismatched, and so you get a negative load score. So that means that the reported father or claimed father is actually not the true parent. So sabi niya kasi this is the tatay, but the software also, if the animal, the true sire is in the database, then they can actually, the software will tell you ito ang tamang tatay. It's not this one. So for this report, actually, it's a negative load score. So that means the reported father is not the true father of this calf. The same with this, 5 mismatches. Normally, we allow 1 or 2 mismatch. That is good because there's actually what we call genotyping error. And you will notice, kahit meron itong dalawang mismatch, positive pa rin. Isang mismatch, positive pa rin. Because it's not just the number of mismatches that actually tells you or increases the confidence. It's the type of markers that went into the selection for the whole set. Because you want markers that have many alleles, like kinaganda kay microsets, 17 number of alleles or forms of the marker, 2 as against 2. I would choose the 17 over 2 because 17 gives you much more confidence. Kasi you will have 1 over 17 chance na yung pareho sila ng size by chance alone. Yung tatay at saka yung putative sire or na animal that's being matched. So that means the calculation for the confidence is actually the probability of the two animals being matched are related to each other gets higher with the set of markers that you choose that are have many alleles. And it's also good to have some markers that have what we call rare alleles, because if the animal happens to have that rare, kukunti, ibig sabihin ng rare is very, very few yung size ng alleles sa population na yun. at na-pick up niya yun. So that means that even if there are two mismatches, but the animal happens to have the rare formatted, then confidence of predicting whether they are really related to each other gets higher. So there are aspects of different aspects of it, or things that we consider when we include which markers actually are going to be put into certain kit. So this one, also like to take note that this what we're using now for buffalo is also we expanded it for cattle as well. So we worked around if we expanded the number of markers from the previous that will work across the two species. So we changed the combinations from the previous original buffalo sets lang at dinagdagan namin. And so the current technology we have right now is possible now for cattle and buffalo. So with a partner laboratory that we have, we're actually now going have the ability to offer this testing service for cattle and buffaloes for those that are interested, the farm owners, the associations that need to have to check whether they actually have the right size or the right pedigree of their calf. It can be made available already. So ito po ang tawag namin dyan is very sire. So it's the first that we can offer. So it's not going to be, it's developed by PCC, but we do have a partner laboratory that will be able to run the testing service, not just for the extraction, but as well as pati na yung report na this is what you reported or you claim to be the true father is not or is really the true father. So up to the point doon na service na gano'n. But sabi natin is that's just on the pedigree side. There are many markers that actually are influencing the performance of a particular animal for certain production traits. For our buffaloes, obviously, we are working on milk production and milk components, fat and protein, those genes that are related to milk production. So bakit magandang gamitin ang DNA marker to complement the effort that we are doing in terms of yung conventional breeding program ng PCC. One is, sabi natin, according to literature, DNA markers are actually advantageous for traits that are lowly heritable. Well, milk is actually moderately low heritability at about 0.25 to 0.3. Sex limited, so we know that only the female of the species of the kalabaw or the baka is actually going to express the milk. So that's limited only to the females. It's expressed late in life because you need to breed them and for them to give birth before they actually express the milk production, which is the interest of trait for dairy buffalo. It's difficult or expensive to measure. Well, it might not be expensive, but remember, 10 months lactation means we have to keep on recording a one lactation period for once a month for 10 months. So that's actually medyo mahirap, hindi lang yung minsanan makolektahan. And it has unfavorable, or if a trait has unfavorable genetic correlations with other traits. So milk actually is, because it's known that milk production is actually negatively correlated with fertility. So as you push for higher milk production, medyo naapektuhan or the fertility traits actually become affected or might have an adverse effect to it. So mapansin nyo lahat halos na sinabi ng literature applies to our kalabaw. Okay, so what else? Okay, so this is the typical program that we have right now sa PCC. First is to identify young bulls for AI. How do we identify? Based on pedigree. Sino yung magandang nanay? Sino magaling bumata? Sino yung tatay? Magaling ba yung pedigree ng tatay? On the basis of that, we pre-select what we call parent average breeding values. Then we collect the semen. Then we inseminate these to our different test stations so that daughters are born out of the individual bulls o young bulls. Grupo yan usually, sabay-sabay silang pinipili. Then only then will the sires have their own breeding values performance because the bulls do not have milk. we have to wait for their daughters to have milk bago namin malaman how good is the individual bulls as a parent. Okay, so that's what we call sire EBVs. Then that's the time we actually put them into the general population or i-disseminate na namin sa various cooperatives that have the dairy buffalo cows. But out of the top rank, we select about 2 to 5% to be mated to bull mothers. These are what we call very good elite cows. Okay, by then it has taken us 8 years, and the bull calves born out of the elite matings between the best bulls and the best mother again goes through the same cycle. Yan. So very long. Sabi namin, how can we shorten it? Well, you cannot shorten the physiological gestation and saka growth period ng mga animals. Ano pa? That's one problem why we resorted to having molecular genetics for the applied breeding. And what else? So we've worked on improving the milk production. This is actually what we call now the genetic trend. Through the years, that's a very long time, pero it's really a positive growth for milk. This is for fat, and this is for protein, milk protein. So we say that, oh yes, we are doing well because the yield of our recorded cows, the ones in the nucleus, the ones that become parents of the next generation are actually improving through the years. Talagang very positive trend. But there's a consequence to it. Diba sabi ko kanina may negative genetic correlation. The consequence is the average fat has been decreasing through the years. That's the negative trend because milk production and fat and protein percentage is actually opposite, negative genetic correlation. And it's very difficult to incorporate and balance selection such that tumaas ang milk, tumaas din ang fat and protein. The yield definitely goes up. You don't know this, but when we plot the percentage, amount of the fat in the milk actually it's going down. So those are the things that actually problem areas where the potential application of molecular genetics comes in, or the use of DNA markers. Other than that, specific genes, actually, SNPs, other DNA markers, can also be used in some aspects. Like these are some lang to. We have like identified specific SNPs from previous projects. This one is screening for the beta-casein, or what we call A1, A2 milk. Okay? So yung beta-casein gene, there are various forms, and we say that the A2 variant of the beta-casein gene is actually favored in terms of digestibility and availability sa humans. Okay? Marbling check is test for marbling in swine, in cattle, or even buffaloes. Prolip pig is a combination of 3 genes that act with fertility. This is only specific for swine. Slick gene is what we call the thermotolerance gene. The GAT1 is for milk fat. This one, LEPR, and LEP is fat deposition. And then there are also other applications of by screening for individual genes or individual DNA markers for genetic defects. So you have halothane genes, frontal hernia, citrullinemia, blood. So these are the individual applications other than yung sabi natin. But I'll get into later bakit yung cycle namin ay napakalaking impact ng ating recent DNA technologies. But these are some of the useful ones that we can use and screen individual animals prior to selection. So ano naging effect? I'm showing you the pig. Bakit? Kasi ang bilis ng response sa pig eh. Ang kalabaw sabi ko kanina 8 years. In pigs you can have like 2 pharaohs in a year. So if you apply in the decision making, in the selection of your breeder sows and breeder boars, you apply the markers. If you know the favorable markers that alleles of the markers that you are interested with, and apply this and select on the basis of that and plan the mating, then you transform the productivity of your herd. So this is like an end of the project wherein we actually screen yung prolific kanina, kasama dito itong ESR. Okay, so we actually screen landrace and large white for ESR gene, and we know that the favorable genotype is 2 copies of the B allele that actually imparts a little bit more piglet per sow per year. That's what the literature said. And so we screened it and validated it on various breeder herds dito sa Pilipinas. And what did we find? Like say, we ran the statistics for it. I'm just going to show you the parity 1 to 6, but we ran the statistics on 1 only muna kasi kukunti na sa ibang record. So the largest number of record is on parity 1. So you will notice AA on the average is 9.6 as compared to the favorable genotype of 11.7. So you immediately have a statistically significant difference. So if your sows are carrying at least 1 copy of the B allele that is favorable, B allele of the ESR or estrogen receptor gene, then you are more likely to have sows that are going to give you at least 1 extra piglet per sow per year. If you're a commercial herd and you have 1,000 sows, and half of them actually will carry the favorable allele, you stand to gain a lot by just having your horse genotype and select the ones that actually are BB genotype. Then you have increase already in terms of productivity. And the same trend follows also for the rest of the parities. It's really more of sabi nila, bakit mas mataas na pagdating dito ng AA? Well, you will notice that the ones with sows with AA genotype actually gets culled more likely. So, ang natitira is yung talagang magaling na lang. So, meron na ditong selection bias. But on the average, the good ones actually stay a bit longer. You get more money out of your placement or your sows. So, that's the practical application of having genotype information prior to selection of your breeders. Yan. So, hindi lang yon. Kasama kasi doon sa fertility is ng ESR is PRLR and the leaf gene. So, you will notice for the PRLR, its main effect is not on the number of piglets, extra piglets, but actually on the ability, you know, the PRLR is prolactin. So, it's the ability of the dam genetically to nurture the piglets up to weaning. And you will notice again that the ones that actually have the favorable allele is the one that actually statistically higher or mas mataas ang weaning rate. Ibig sabihin, they were able to nurture yung live piglets nila to weaning more successfully than the ones that actually do not have the favorable allele or in the favorable genotype. Yan. So sa dalawa, hindi yun ang main effect. Okay, I'm sorry. Am I not coming through clearly? Hello? Am I okay?
Yes, Doc. Opo.
Okay. Sorry. Akala ko kasi medyo okay, thank you. So this is you will notice and what happens after 3 years, sabi ko mabilis if you just keep on applying the right decisions by genotyping future breeders. What happens to the population? Sabi ko kanina, what we want with animal breeding is the shift in the frequencies of the favorable so 3 years after, we started with the screening in 2015. At the end of that, when different participating farms have started using that and selected their boars and used the boars that actually have the ESR, what did we see? So the green ones for the ESR actually from 2.2% at the start, because selection then for the large white and the large white and the ano may kasama ni large white yung ating maternal lines ng pigs land race pala, sorry, yung dalawang yun are what we call maternal lines. It was very difficult for us to find the BB genotype because they were selecting on the basis of ADG. Hindi naman dapat, kung maternal line kayo, you should be selecting on fertility. Sabi na, bakit hindi nag-i-improve ang overall average ng Philippines noon, ang average na number of piglets born per sow per year? Is because they are selecting a different criteria, but they wanted improvement in fertility. So I showed them 2.2%. Kaya pala. So when we started that, and wala pang BB noon, konti lang. Slowly, when we mated the heterozygous, the AB, with identified AB rhina females, eventually they were able to produce the BB and sold the semen of this to other mga clientele sila. So the population, the frequencies actually increased to 13.9 after 3 years. So there was immediate effect just by using the right technology on the right kind of traits as well. So ganun din na nakita natin sa apat kasi yan na group of genes that are associated with fertility. RBP4 is for the semen quality. So bakit ano lang bumaba siguro is bakit red siya? Bumaba ang leaf. Okay? Pati yung proportion niya, medyo bumaba lang ng konti. So maybe nabigyan kasi na masyado ng pansin siguro ito kesa sa leukocyte inhibitory factor or the leaf gene. Kaya ganun. Pero napakalaki ng jump sa ESR kasi itong pinakamalaking major gene in fertility in swine. So can you imagine that in a matter of 3 years, there was already a big change, a shift in the population. So, sabi namin, sige, paano natin gamitin ngayon yung DNA technology para ma-shorten natin yung problema ng dairy cattle na it takes us 8 years to know which are the best bulls for breeding. So, we did, other than yung mga individual screening, is actually the best one to apply is what we call genomic selection to apply to the breeding program. So, we cut down from 8 years to 3 years. Why? We shorten it. When we actually do genomic selection, we don't wait for the bulls to have their daughters born to be able to know their genetic merit. We identify on the basis of the individual genes that they carry or SNP markers that they carry, and each one has a contribution to the whole of the milk production as a trait. And this is summed up, and the credit for that is what we use to rank them individually. So the ones that are actually mataas by prediction become bull mothers immediately. So from 7, we shorten it to 3, and they produce the calves again without we bypass the progeny testing. So that's the promise, and that's the potential for genomic selection by shortening the selection process. We cannot shorten the gestation length, physiologically, but the selection process we can shorten by not waiting for the daughters to be born and use them immediately on the basis of genomic prediction. So why genomic selection? Well, mas accurate. Why? I'll tell you later. Cost to produce AI bulls is significant. So by shortening it, the bulls stay in the AI station shorter period of time. We save on cost. Bakit daw? Mas mataas ang reliability ng genomic breeding values kesa yung sinasabi ko kanina, pedigree-based or parent average breeding value. So reliability is just the square root of accuracy, if I'm not mistaken, but the reliability increases with the use of genomic information. Breeding values can be estimated based on 1,000 markers instead of own performance or family information using pedigree. And you can actually get the breeding values early kesa late. So yun ang promise and potential of genomic selection, which we actually like. Furthermore, hindi kasi namin fully magamit ang progeny testing sa setup ngayon because we actually can test very limited number of young bulls with a small number of daughters with lactation record per 2 Yun ang one. And sabi ko, 8 years, very slow. So those are inherent constraints within the PCC other than yung mga sinasabi pang iba kanina. So we went through the availability of what we call the buffalo SNP chip. This is a chip with 90,000 SNP markers distributed throughout the whole genome. So it has 123,000 probes representing 90,000 SNP markers. Whole genome, that's why we call it genomic selection, because we make the prediction on the basis of these 90,000 SNP markers. Yan. It covers the whole genome of the buffalo with 21.6 kb distance. Yan. So, base pairs niya is 2.658b per billion. Okay, so konti sa cattle. Okay, so, but before we proceed, there were some preliminary information that we needed to see. We have several breeds available. Can we consider them as one? So we needed to see the population structure. But actually, out of the 90,000, how many are usable? When we say usable is meron siyang variation. That means hindi siya homozygous, heterozygous yung marker na yun. So meron AA, AB, BB. So ganon. So we have to check all the 90,000 markers and say which ones are actually usable. Is it dense enough? Are the number that are heterozygous or what we call polymorphic, I'm sorry, is the number of markers polymorphic sufficient, dense enough? Marami ba? Baka naman puro mga homozygous yung mga SNPs na yon. So we needed those preliminary information, and then we have to decide ano ba ang reference population namin. The next is, are these associated with no production traits that we can use for breeding program? So we ran through that, including genome-wide association study. And we found out there are actually, yes, significant markers that are associated with milk production. So we tested different breeds, including swamp buffalo. We used the action analysis with software to generate the genotype call. So the software gives us from 0, 1, 2. And we use that, yun ang raw data. So the 0, 1, 2 goes into R and set the default settings. May mga call rates that are less than 97%. So pinadrop yun. So out of that, we actually get about 70 something. So the rest of the 75,000 polymorphic and usable SNP markers we use now for downstream analysis to create what we call genomic relationship matrix, principal component analysis to be able to visualize the population structure. How are the different breeds that we have right now? Si Brazilian, Bulgarian Mura, si Swamp Buffalo, how are they in terms of structuring? So we wanted to visualize that, and then we wanted to see, are the SNP markers, is there a usable linkage to equilibrium? Are they linked to each other such that we can use them for prediction? So we use Blink software for that. Our package that is actually linked to the Blink. Okay, so we wanted to find out if there were actually really SNP markers that are associated with production. We have already prior report of that, but we wanted to see that. So we did a GWAS. So GWAS 65,680 SNPs were used. And we consider p-value 0.0001 to be significant. So ito yung different breeds. Tatlong klaseng riverine at isang swamp that are polymorphic. So swamp, maraming homozygous sa swamp. So hindi talaga. So in terms of the one we want for the dairy buffaloes is we use actually we combine the Brazilian and the BMP. Ito yan. And some of the American Mura na dito. Ito yung team. These are the Italian. They cluster separately. And this is the swamp ang layo. So, we cannot consider the Italian to be clustering together with the rest of the Euro-based breeds. So, this became our reference population for development of genomic prediction models. So, this is actually what we wanted. Reference population means the females that have the milk production. So we genotype close to 2,000, but the ones that are actually usable is close to 900 animals. And on the basis of that, because we wanted to see individual SNPs have, sabi ko nga, di ba may favorable at saka not favorable kasi ang SNP is only biallelic. Dalawang form lang siya. So, on the basis of estimating the SNP effect for each of the 65,000 polymorphic or usable SNPs, we estimated the SNP effect for each of those, and a prediction is to sum all of the effect of individual animals. So an animal might have like 90% are favorable, so taas ng credit ng kanyang value, no? As compared to an animal that only has like 25% carried ang kanyang favorable. So, on the basis of that, we do a genomic prediction. The idea is, if we know kung anong klaseng SNPs allele, is it the A or the B, is favorable and each one has an effect and we sum it up, we look now on the young bulls. Wala silang gatas, pero they carry the same SNPs. And we actually credit the individual SNPs, target set, DNA tested, walang production yan. That's what we call genomic prediction. And these are where we apply to the young bulls. So we shorten from 8 years to about 3 years or 3.5. So we actually save cost. Yeah. So that's essentially what we have in terms of program of applying the genomic information to our selection of the future bulls. Yan. So, ito yung females. We apply the 90K. We have the prediction equation. We genotype potentially young bulls. On the basis of the prediction equation, we rank them, the selection candidates, and rank based on genomic breeding values. The top ones actually go into the breeding. So, essentially, that's what we call genomic selection. So, the advantage ng SSG blab, single-step G blab, is what we call how we do the evaluation for the genomic prediction as compared to the pedigree-based lab for testing and prediction. So, ito yung dating ginagawa kung may EBVs. So, how do we compare the conventional one, 8 years namin na ginagawa na cycle as compared now for the SSG blab model for genomic selection or genomic prediction? So, mas mataas ang accuracy ng paggamit ng genomic ng single-step G blab compared sa pedigree-based blab. In terms of yield, we find the same thing for protein yield and for fat yield. So, hindi lang naman kasi sa milk yield namin in-apply ang prediction. Yeah. So, ano pa? It actually corrects also pedigree. Bakit? Ito yung A matrix is the conventional one, pedigree-based namin ng numerator relationship matrix. So, an animal is related to itself is 1. An animal that is related to an anak is actually 0.25. So, we might say that 2067 might be the father of this kasi 0.25 relationship. Ito rin probably. So, sila ay half sims. So, you'll notice directly lang ang pagkukompute ng family relationship. But, with genomic relationship, by doing the genomic relationship matrix, you actually extend yung relationship sa iba pa niyang kasama because you are not tied down by the information on the pedigree alone, but let the genes speak how related they are to each other within the population. So, within a family, we genotype some, the other one is not dependent. So, normally, A matrix lang ang ginagamit namin dito. So, ito lang ang alam namin related. Hindi sila related. But if you genotype the other ones as well, yan,
magkakaroon ka na ngayon ng relationship with the other family, which you normally will not have with just recording the pedigree. So the genomic relationship matrix links the different families. So now we have a very nice relationship rather than 0-0. And that is, it corrects pa rin yung mali because it's not dependent on the pedigree, it's not what you report. It's what the genes are actually telling that these two animals are actually related. So it corrects pedigree error. That's why that alone increases already the accuracy of our prediction compared to the conventional pedigree. But it's also very correlation between the conventional way, ito yun, and the new one, which is yung genomic prediction, is actually 0.84, which is high. Sabihin, by selection naman, we're only interested in this aspect, yung top rank animals. And kung ano yung mataas dito, siya rin yung mataas, 62%, because some of the high ones here actually might have pedigree errors na correct ngayon. Kaya hindi siya 100 na single step gene lang. Kaya hindi siya sama sa top. So that alone gives us better confidence. Pag sinabi niya tama to, mataas din or good genetics yung individual. So within the females then, ayan, nagkakaroon ka ng overlap, but at least the good ones are really on top, whether you do it by pedigree block or by single step G block. Yan. So, yun. So, ito yung isa example ng re-ranking. Ito yung conventional. So, si Warren was actually 494. But when we ran the genomic prediction and gave the assigned or predicted the genomic predict values for the individual young bulls on selection, is 102 lang pala siya. Bababa siya sa ranking. Pero nandun pa rin siya sa taas because 102 is still positive. So there was just a ranking, but yes, definitely the good ones identified by our conventional are still really on the top, except that the ranking will have a change. And these are now our latest genomically tested bulls. Yung pinakita ko kanina, those are the top 6. We actually did not choose the ones that have lower values. These are the ones that are top. So these are the very first animals that we are reporting to be where we apply to what we call the genomic prediction or the program of genomic selection. So these are Lucas and Donald are the first two genomically tested bulls. We have Dexter. Wala lang picture ng dalawa pa because they are young, but we will hope to continue the program year in, year out, and increase the database for our reference population. So future plans, we increase reference population, genomic predictions for other traits that are also going to benefit much with the use of DNA marker technology, especially fertility traits, because these are really lowly heritable. So malaking pakinabang ang paggamit ng genomic selection sa fertility traits. And probably a low density SNP chip designed for local. And also I'd like to encourage you to visit the site. We actually put online the site directory. Yung pinakita ko kaninang sila Dexter, sila Donald is already can be viewed dito sa site directory. This is the site so that you have more information with regards to our semen tools. Yeah. So that's it. Thank you. So I'd like to acknowledge a lot of the brain work was done by Romel. And the OSTP card actually for funding the project so that we are able to establish. And that's it. Thank you. Okay, so
Yes. Hello po.
yes. Yes.
Thank you. Thank you po, Dr. Esther B. Flores, for your very informative presentation on the application of genetic markers and genomic information in animal breeding. Now, before we proceed to the second speaker into this webinar session, as I am said earlier, you may send your comments and questions to the chat box And please remember to indicate to whom addressing your question to. And we will be having our open forum after the second presentation. So Doc Ping, standby lang po tayo dyan at mamaya pa po yung ating Q&A after the second presentation.
I'll change channel, no? From the laptop, dilipat ko sa cellphone kasi.
Okay, okay lang po, Doc.
But I'll be online pa rin. Or I'll be just here at the background just in case mag-start yung ating QA or what.
Yes, Doc.
Session.
Apo.
I'll be available. Okay, thank you.
Okay, thank you so much, Doc. And I would like to acknowledge po yung attendance ng ating mga newly signed in na participants from different government agencies and academic institution. Maraming salamat po sa oras na binigyan nyo sa amin to join us. Now, hindi ko na po papatagalin at ibibigay na po namin sa inyo yung ating pong pangalawang presentation. Our second presenter is Dr. Yoprosina D. Atabay. She pursued her college education at CLSU. She obtained bachelor's degree in animal husbandry in 1984 and Doctor of Veterinary Medicine in 1986. Afterwards, she pursued graduate studies at Hokkaido University, Sapporo, Japan, under the Japanese Ministry of Education, Culture, Science, and Sports Scholarship Program. She obtained her Doctor of Philosophy and Veterinary Medicine in 2002. Dr. Atabay took a postdoctoral study at the Center of Regenerative Biotechnology at the University of Connecticut, USA in 2008. She was likewise awarded the Fulbright Advanced Research Scholarship Program at the University of Massachusetts, USA in 2012. She was conferred the rank of Scientist 1 under the Scientific Career System of the DOST in June 2014. And Dr. Atabay is a prolific researcher who has authored and co-authored numerous scientific publications in animal reproductive biotechnology and reproduction, with water buffaloes as the main animal commodity. And Dr. Atabay's dedication in her field has earned her several scientific awards. She is currently the Supervising Science Research Specialist and the Officer in charge of Research Development Division here at Philippine Carabao Center, National Headquarters, and Chinpool. Ladies and gentlemen, it's my pleasure to introduce to you Dr. Yoprosina de la Peña Atabay.
A pleasant and a blessed day to everyone, and thank you so much for that very generous introduction. So again, welcome to all the participants of this webinar, and both from various research and academic institutions. So for my topic this morning, actually, this is entitled Advances in Innovations in Livestock Biotechnology. And basically, this is for food production and resiliency. So again, good morning and welcome to my presentation. And okay, the title is, this is basically our sharing of the advances in innovations in livestock reproduction for food production and resiliency. And for the outline of my presentation, this is very much in complement with the presentation done by Dr. Flores, which is on the breeding and selection. And it is just but proper and right to start with the selection of best animals, because our function or work then is to be able to reproduce or multiply these genetically superior buffaloes or any other livestock through the reproductive biotechnologies. And that is my emphasis for this morning. And of course, there's no better way to start or to focus but on the uh, biotechnology that is used by the Philippine Carabao Center in the upgrading and propagation of our buffaloes. So I am referring to the artificial insemination, which had been used for almost 30 years. And of course, what are the recent developments with regards to artificial insemination? And we call it the fixed time AI, which just entered in 2014. And then there are subsequent to that are the modification of the fixed time AI. Now we have the enhanced AI and the select AI, which are made to be able to made it simple and doable by our artificial inseminators or field technician. And my presentation as well will cover what are exciting developments in livestock reproduction. And this would include the semen sexing technology and how do we assure that that the desired sex of the calf or the offspring that we produce. And because this is very much relevant if we need to produce dairy animals or we would like to choose the meat animals. And also other exciting developments in the the technology on we call the multiple ovulation and embryo transfer. So this basically are addressing towards our current drive or program by the Department of Agriculture towards increasing the productivity and profitability from livestock farming. And of course, before I go to the proper topic, allow me first to introduce my workplace, the Philippine Carabao Center, which is an attached agency, the Department of Agriculture, and it is created by the virtue of Republic Act 7307, otherwise known as the Philippine Carabao Act of 1992. And our main mandate is to conserve, propagate, and promote water buffalo as important source of draft power, milk, meat, and hides. And this is towards the to improve the general well-being of the rural farming communities. So ultimately, our aim is to be able to establish a buffalo-based enterprise to provide additional income to our farmers. And to accomplish this mission, we have the Carabao Development Program. And I would like to emphasize that this is a social program, and it has three main components: genetic improvement program, Carabao-based enterprise development, and the research and development. And if I may emphasize, genetic improvement is covering or dealing with the animals, with buffalo, which is our medium for service to our clients. And the second one is the Carabao-based enterprise, which is the human or the social aspect of our program. And of course, I would like to emphasize the research and development aspect of the Carabao Development Program, which enhances our the two programs, the genetic improvement and the Carabao Development Program. So this is encompassing actually and even the heart of our Carabao Development Program, the research. So allow me to present what are our research agenda. And our main strategy in research is R4D, so research for development. And this includes the following disciplines: animal breeding and genomics, reproduction and physiology, biosafety and environment, production system and nutrition, and also product development. And okay, just to, our main research focus is first, is the transformation of the Carabao as draft animal. Traditionally, it's a draft animal to become a source of meat and milk. So being a draft animal, the carabao can only produce around 3 liters of milk. And it is, as has been described, it is a very long process. But okay. And then another objective for in our research is, as I mentioned, is to be able to multiply these genetically superior animals. But there is a main challenge that we are facing right now. We have to increase, of course, the local milk production. And of course, in order to do that, we must be able to increase, first and foremost, the number of dairy animals in the country. And however, buffaloes especially is being faced with a great problem, or it could be intrinsic or extrinsic. So basically, they have delayed sexual maturity, they are exhibiting seasonal infertility, and then they show poor estrus manifestation, and there is a problem of asynchrony in timing. And these are resulting to low conception rate, long covering interval, and low reproduction efficiency. So having said all these problems in buffalo reproduction, there is a need for us to come up with an effective production management system. And this includes nutrition, animal health, breeding, and for emphasis, our program in reproduction, in order for us to make all these outcomes in terms of production, productivity, and profitability higher or more efficient. So in line with that, we have developed an intensified reproductive management program for buffaloes. I think this was in 2014 or earlier. And we call it the Triple E strategy. And it stands for enhancing pregnancy rate, early pregnancy diagnosis, effective rebreeding program. So this is a cyclic event. And all this strategy are all anchored on our clear understanding on ovarian physiology. So in terms of enhancing pregnancy rate, we have all these techniques. I mentioned the fixed time, the in vitro embryo production. And with regards to early pregnancy diagnosis, to reduce the calving interval and make animal more productive, we have the ultrasound and other early pregnancy diagnostics. And of course, after determining that the animal is not pregnant, it is important to put them back to the rebreeding, to the herd for rebreeding. So this is the Triple E strategy. So now allow me to go to the main technology that we are using for carabao upgrading, national upgrading program. So I'm referring to the artificial insemination, which is the reproductive tool in the dissemination of superior genetics of paternal origin. And this is the widely used technology. So again, to emphasize, buffaloes are silent heat and difficult to exhibiting a problem in estrus detection. So there is a need for us to come up with a technology that eliminates the need for heat detection and can address the problem in the breeding seasonality, which is very marked under our situation. So just to highlight the difference between our artificial insemination and then the modification or advancement, the fixed time AI, the insemination or impregnation done with regards to the conventional AI, it's based on estrus. So the main difference with fixed time, it is based on ovulation. And then with conventional AI, there is really a need for heat detection, and this would require additional labor. to detect the animal in estrus. But in fixed time, this is no longer needed. And hormone, of course, with AI, is a very simple one. Only prostaglandin is used. But in fixed time AI, there are really combinations of various hormones. And since a very important factor to consider is the ovulation. Now, it's in conventional AI, it is more variable, unpredictable, but in fixed time AI, it's quite tight or close. And the basis of, again, for insemination is estrus. But I think obvious or drawback of the fixed time AI technology is it's kind of costly because it involves a lot of or various hormones. So just to give you the difference between in terms of the approach of the AI, conventional AI. So this is just a 3-day program. So in day 0, the prostaglandin injection is done. And this is to regress the semen and allow the growth of follicle. And after 3 days, the animal is expected to go in estrus. So you do the artificial insemination. And this is done at 8 hours interval. So that is just the simple AI. And in contrast to the fixed time AI protocol, it aside from the use of prostaglandin, it involves the use of ovulatory hormone. We call it the gonadotropin releasing hormone, given 7 days before prostaglandin and 2 days after prostaglandin injection. So this would make the ovulation very precise. So, and then the artificial insemination is done fixed 16 hours after the second generate. So normally AI is done before the expected ovulation. So that's the fixed time AI. So we have optimized the fixed time AI protocol. And actually it started in PCC in 2014 in our desire or objective to address the hard breeder or repeat breeders in the herd. So we started with the conventional or the widely used, globally used protocol for fixed time. This is the ovulatory or OVSING protocol. So this is it was discovered in 1995. And then, of course, in 2014, we adapted it, and then we tried to modify to be able to increase its efficiency. And now we call the protocol the CIDAR sync protocol. And in here, we involve the addition of the progesterone or external progesterone aside from GnRH. So again, it's a fixed time AI. And then, can you go back, please? Could you go back? So, and our ultimate protocol now is we call the CIDAR Sync HCG because we improve also in what particular ovulatory hormone that will be giving the most the highest pregnancy. So this is it. So if you compare the different protocols, so it's increasing from with OVSINC, we are only getting 31% pregnancy rate, and with the CIDAR Sync GnRH 43, and with our final protocol CIDAR Sync HCG or LH, we are getting around 58% efficiency. So this is interesting. This is as you with ovulation time narrowing, the efficiency is getting high. This refers to the closed ovulation time and gives, this is the main reason for a high efficiency. And then in 2017, the fixed time AI has been commercialized in the country and with the coming in of a private company. So now it's getting around the country, fixed time AI. And this is just the associated activities. Of course, one major difference is we involve the use of the ultrasound in detecting the ovarian status and before we inject or administer the hormones. And in the process, we are assessing the follicular growth and development because it's really important. So again, we do ultrasonography for follicle assessment and monitoring. And at the end, here is the animal showing or manifesting the estrus with all the external signs observed. So following the fixed time AI, because we want to make it more available to our technicians, we developed this enhanced AI. So if you go back to our our conventional AI, this is actually a hybrid of the conventional AI and fixed time AI. In here, during the first AI on the third day, we are adding the ovulatory hormone. So this will facilitate not only the estrus, but even the ovulation, but a little bit not so costly. And in the efficiency is just like tantamount to the OBSYNC or the basic technology. And I also would like to emphasize that this enhanced AI, okay, can I just go through my presentation, sir? So now the enhanced AI is, this is our COVID-19 responsive protocol because, you know, normally when we do artificial insemination in the field, we have to gather people, the farmers and the animals, but it has to be avoided during this pandemic. So we do this, we allow our village-based technicians to just perform this in the field. So, and yeah, so that's for the enhanced AI. So before I go further, allow me to just emphasize the main difference between the cattle and buffalo reproduction. So here are the ovaries or the structures. Morphologically, these are the cattle ovaries. You see, they look healthier, bigger, shining, and yeah, these are the follicles. And see, look at the buffaloes. Almost, this is a natural thing. Almost no structure. And here are the corpus luteum inside. We call it the yellow bodies. It is more marked in cattle. And so that is in terms of the structure. And then another difference is that in cattle at birth, it has 100,000 of follicles or the eggs, and in buffalo it has only 10,000. And so sa simula pa lang parang ano na pinagkaita na yung ating buffalo. And this is the reproductive cells throughout the life of the animal. And in terms nga of ovary ay talagang small. And then of course the corpus luteum, which is have a big implication in pregnancy, is also small. So also I would like to emphasize that ang cattle, it produces offspring every year kasi the gestation period is about 280. Whereas in buffalo, twice every 3 years because it has long gestation period. So mas medyo mabagal talaga ang buffalo. And then, yes, okay. again, just to, as I was, this is also a product of our intense research in buffalo reproduction. So just a simple background on the buffalo estrus cycle. Yes, there is 21 days estrus cycle of the buffalo, and the cycle has the luteal phase. This is the corpus luteum. It's luteal phase, which is quite longer, and then the follicular phase, which is where the, yes. So, and then throughout the 21-day cycle, we have the three waves, follicular waves, 1, 2, and 3. But these waves are not, they can produce dominant follicle, but they are not ovulating. So there are also animals that are also two waves. So this is a two-wave cycle animal. And only the last wave will ovulate. So this will also determine the success of AI. So if you are doing AI during this wave, two waves, then you're not getting the pregnancy. And this is also the presence of corpus luteum that produces the progesterone. That is why there is no ovulation happening. But during follicular phase, when all this inhibitory effect of progesterone, there is already the successful ovulation. That's why in fixed time AI, when you induce the estrus, you are able to eliminate those unnecessary waves. You go directly to the a phase that are able to produce the ovulatory or follicles. So this complicates the efficiency in buffalo pregnancy. Yes, sir. Okay, also in our research through many years, we have confirmed indeed that in reproduction, size matters. So, but specifically, I'm referring to the size of the follicle, which is when you know, at the time of artificial insemination, or when the technician is ready to inseminate the animal, it is important for us to achieve the optimum size of the follicle. So because if it is small, remember that the follicle contains the oocyte, it contains the different hormones, and it also this is also responsible in the formation of the corpus luteum, which is responsible for the maintenance of pregnancy. So the optimum size should be achieved, because if it is also too large, then the oocyte inside is aged, and that would result to lower efficiency. So optimum size matters in reproduction. And also, this is one recent development or protocol that we made, which is for practically for our AI technicians, as providing them the guide. So I have been emphasizing that the follicle size or the structures in the ovary is very important. Now, the AI technician does not have the ultrasound for them to be able to detect all those structures, but they can palpate. So based on the palpable corpus luteum or palpable, so based on the palpable corpus luteum, they can start to select. So we only have to select those animals with CL. And then based on the follicle size, they should be able to do the insemination. So it is important that the size of the follicle should be, again, be determined for timing of AI. So again, this protocol is designed for a practical application in the field based on the structures of the ovary. So we have to train more of our technicians to be able to to determine all these structures, but they are palpable. So these are just the associated technologies. When we do the fixed time AI, I've said that we use the ultrasonography for detect whether the animal is pregnant or not. And then it is by ultrasound, we can detect as early as 30 days if the above animal is pregnant. And then, of course, the recent development, we the pro or associated pregnancy associated glycoprotein test, which can detect even earlier pregnancy in buffaloes. And our final confirmation is again the rectal palpation at 60 days. So this is how we. This is just to emphasize that the pregnancy-associated glycoprotein is now becoming a pregnancy kit available commercially, but we have not yet done it here. But we use rather an ELISA technique in the laboratory, and we are accepting samples for. So essentially, the tandem of fixed time AI and the PAG technology is the most desired technique nowadays in reproduction, especially in advancing countries. Okay, oh, this is just the basic thing also to confirm that the animal is pregnant. We measure the progesterone. So progesterone is produced by, is a hormone that maintains pregnancy, and this is produced by the corpus luteum. And we are able to determine that in the milk as well as in the blood. So in here, if the animal is pregnant, obviously it is a higher progesterone concentration than the non-pregnant animals. So here are just the calves or that we have produced through our fixed time activities nationwide. Yeah. We have done it in Luzon, Visayas, and Mindanao. And this is also, you know, when you do fixed time AI, you are able to come up with animals in contemporary, meaning they are in group. And we also do that in cattle. So these are the very first born calves from fixed time AI. And then, yeah, I just want to emphasize that our project in the fixed time AI and other advanced technologies were made possible through the funding of the PCARD or UST PCARD. And this is a bundled project actually, wherein the reproduction is just one of the components. It is a recent three-year project that was recently completed. Yeah. Well, I just want to mention the other innovations and exciting developments which could be a potential, actually could even boost, especially the dairy industry. This is the use of the semen sexing technology. So as of now, this is a patented technology. And this is the use of the sexing machine. So in here, this enables the separation of the Y chromosome bearing sperm and X chromosome bearing sperm. So those with, of course, X chromosome bearing sperm can be used for artificial insemination to produce dairy animals, and the Y chromosome bearing sperm can be used for beef production. So this is a patented technology. And now we are in collaboration with NDA, who purchased the semen, sex semen of cattle or dairy cattle, and we are using it for fixed time AI. So it also in tandem with the the sexing technology, of course, your ability to determine the sex of the calf. So just like in human, we are excited to know what is the sex of the fetus. So, and here we were able to, what are the structures and, yes, basically this is the genital tubercle, which give rise to the female genitalia. So when this is seen, and then it's a girl. On the other hand, we are able to determine if the sex of the fetus is a boy. So that goes hand in hand with the semen sexing technology. And as I mentioned, the problem, the project with NDA, we were now able to produce a pregnant, or confirm that it's really a female, using the semen, the sex semen. Okay. So another recent development or completed project, this is funded by Picard, is the multiple ovulation and embryo transfer. So this is multiple ovulation. So if we have the fixed time AI, it deals with a single ovulation only. In here we want to produce more embryos, more offspring from genetically superior animals. So we are referring to a donor, and then we inject or administer the hormones for super ovulation or multiple ovulation. And then again, the fixed time AI is involved. And then we do the embryo flashing, embryo searching, and all the works in the laboratory. So basically, it's like, these are injection of the super ovulatory hormones. And then this is done during the collection of the embryos in the field. Here are the flash embryos, researches in the laboratory, and these are the in vivo derived embryos. And yes, we are also exporting under our project, and one embryo is costing 25,000. So if you will value this, I don't know how much do we have. And then, of course, we are storing it as a frozen embryos, and later we do the embryo transfer. So the embryo transfer also, while we have the donor animals, we also have the recipient animals. Again, we do the fixed time AI, but then we do the hormonal injection, and then we do not do the AI, but we transfer instead the embryos. And so this is our the result, the major result of okay. So this is just the fixed time. Now we initially we have the fixed time AI, and in here we are introducing the fixed time embryo transfer. So basically it's the same with fixed time AI, but in here, as I mentioned, instead of doing AI at the end of the treatment, we transfer the embryo. So it's called fixed time embryo transfer. So I believe this is the major development in the multiple ovulation and embryo transfer technology. And we are able to establish that here in the Philippines, and also the production of embryos. And okay, I am so happy to share with you the Pag-asa, which is the very first fixed time embryo calf ever produced in the country. And we also call him COVID calf because he was born in the height of the extension of, the ECQ. That was April 24. So it weighs 32 kilograms, it's bouncing and healthy. And now he is 5 months old. So that is Pag-asa. Okay, so other important development that I would like to mention is on the mammalian fertility. And this is what we call the PLC theta. So before we in the mammalian sperm, we only identify it as a sperm factor. But in the recent year, it was known to be called as the uh, phospholipase C theta. So we are able to demonstrate it indeed in the laboratory. Sorry, let me just finish. I'm finishing up. So this is just to demonstrate and to prove that the buffalo semen has this PLC theta. This is the fertilizing factor. And we are now, we have developed a technique for which involves the RT-PCR. So yes, it's basically involved in the activation of the oocyte during in vitro fertilization, and then finally for embryo development. And we are using this technique for the production of embryos, both cattle and buffalo in vitro, and to ensure that we are really producing superior and fertile embryos for embryo transfer. So anyway, just to recap my presentation, I would like to emphasize that the reproductive biotechnologies are important tools for genetic improvement as well as the multiplication of buffaloes. And FTAI is focuses on ovulation, achieving a higher efficiency. And then we develop an enhanced AI, which is doable and cheaper for our technician. And then also the select AI, which also the recent protocol that we have produced based on the structures. And also, so I would like to emphasize that the understanding on the ovarian physiology, endocrinology, and fertility would really allowed us to achieve higher efficiency. And of course, the exciting developments are innovations in the field of semen sexing and multiple ovulations. These are the most recent ones, and they have a potential for commercialization. So what are our plans? Yes, of course, we want to work on the deployment of this technology for our clients and for, of course, for the commercialization of possible technology. And actually right now we are collaborating with Dr. Flores to come up with a service that would use a genetic superior semen and then with our fixed-time AI technology. So also I would like to emphasize that in general, the PCC is doing research for development with direct application to our farmers, and then technologies and innovation for the livestock industry, as well as for the benefit of our farmers. So lastly, I would like to thank, yes, ultimately, we are working, our research and development efforts are geared towards for the benefit of the rural farming community. And lastly, I would like to thank the, yes, of course, the Picard for all the supporting our program in reproduction, as well as, of course, the Biotechnology Program Office for their continued support in advancing the state of our program. And of course, the LBC for sponsoring this webinar. Again, everyone, good morning and thank you for your time for listening. Thank you.
All right. Thank you, Doc, Bing Atabay, for the very informative presentation on the advances and innovations in livestock reproduction for food production and resiliency. Now, without further ado, let us proceed to our open forum. You can now ask questions to our speakers. And yeah, it looks like we have a few questions. So our first questions is for Doc Bing Flores. Hello po, Doc Flores. Hello po, Doc. Okay, so wala pa po si Doc Flores. So yeah, we have a question to Dr. Atabay. So the question is from Sir Joel Salazar. Okay, so okay po. Doc Atabay, do we have a national production standard for buffaloes for us to know if the reproductive performance of buffaloes are efficient?
In terms of reproductive efficiency, through the years we were able to determine the efficiency that we are getting from our services. For example, our artificial insemination. With regards to our AI program, we are achieving around 25% efficiency. And then with fixed time AI, we are able to raise that to 40%. Yeah, so around that standard.
Yes.
And
Yes. We have a follow-up questions also from Sir Joel Salazar from Pampanga State Agricultural University. And this question is addressed to our both speakers, Dr. Flores and Dr. Atabay. So based on your scientific experiences, which of the two dairy animals, the cattle and buffalo, is best to raise to help in increasing our milk production? Sige po, Doc Bing Atabay, kayo na po muna ang maunang sumagot.
yeah, of course, in terms of the ability of to produce milk, of course, it's the...
Compute nyo lang, sasagutin ko lang po.
Okay, sige. Practically, in terms of the ability to produce milk, as what I've said, as of now, we are reaching to the maximum of around 7 or 8. So, compared to cattle, so it's really a dairy animal, the cattle, and buffalo can only support. That is why it is a main support to the dairy industry. And but again, through the years, our contribution to the dairy or production is getting higher and higher. But the cattle is contributing around 65% of the total milk production. But the buffalo is only around 36. But it's getting higher and higher because of our genetic improvement program.
Hello po, Doc Bing Flores po. Nandiyan na po kayo?
Yeah. I'm here. Sorry,
Okay,
kanina
so,
na yung mic.
Opo.
Okay, so to answer the question, tama
Yes
ba?
po, Doc. Opo. Go ahead po, Doc.
Oo. Actually, ang sagot ko dyan is not as straightforward as yung which one is better. It's more of ano yung management conditions at saka yung resources available. If you are going into medyo intensive yung talagang commercial, and then I would say that you can put up all the necessary inputs, then you can go opt for dairy cattle. But if your system is, let's say, gaya ng clientele natin, small holder, you have only kukunti ang resources, then I guess the kalabaw will be more applicable in such situation. Yun. So, it depends, kung maliit ang resources mo, not so much input, actually, mas maganda ang kalabaw. Lalo na if you cannot provide yung yung housing that is appropriate para kay dairy cattle, and the kalabaw will more likely be a better option for you. But it doesn't mean naman porque mas mababa ang production niya is mas kukontinti kitain. In fact, if you have a low input and you look at the price difference per liter ng cattle and buffalo milk, actually, halos doble ang difference nun. So if you're producing 10 liters of cattle milk and you're producing about 7 ng buffalo, it times 2 mo yun. It's actually, you end up earning more per liter pag sa kalabaw yes, sa dairy cattle. So it depends on your situation. We can give you a straight answer for that.
Okay, thank you so much po sa ating dalawang speaker sa pagsagot po sa question ni Sir Joel Salazar. Okay, so our next question po is for Doc Bing Flores po. Okay, so ang sabi po dito, this is a question from Sir Anon to Doc Flores. Will the breeding program affect the genetic conservation of Philippine Carabao?
Actually, no, because the program I showed you is only intended for dairy buffalo. So we also have what we call a separate program of conservation for the swamp. We what is not really actually na ipapakita palagi is we actually have what we call herds din na swamp buffalo to maintain, and we actually have on hand din lagi always available semen from our swamp buffalo that has a separate breeding program as well. So we're not actually disregarding the swamp. In fact, we put in place a very good breeding program based on growth rate and conformation para ma-sustain pa rin natin yung talagang distinct advantage ng swamp for draft and a source of milk. So we have that. You only need to request, yun na lagi ko sinasabi sa aming mga AI coordinators is we always have semen, frozen semen available from our swamp buffaloes that has also undergone the same evaluation but for a different rate, which is yung growth. I also would like to encourage you to visit our online site because there you will see yung pictures ng mga swamp buffalo bulls natin available. So I hope that answers your question.
Okay po, thank you so much po, Doc Flores and Sir Anon. And our next question din po ay from Ma'am Delive Ramos Tabon from Benguet State University. And this question is for Doc Flores again. So Doc Flores, what practical or economical breeding program can we implement?
Or do you need
to implement to improve the growth rate of native animals? Hello po, Doc Bing Flores.
Mentioned earlier po, as mentioned earlier, ang selection namin for swamp is really basically based on performance in terms of growth rate. Nung ni-review ko nga yung performance nila in terms of average daily gain, the selected ones actually is mas mataas na compared noon. So, we do not want a very big, large frame kalabaw kasi baka kalad ka rin kalabaw yung ating farmer. But we want them growing faster. An additional trait actually na idinalagdag namin ngayon is what we call scanning for eye muscle area. Ito yung ginagawang yung cut ng para sa rib eye. Bakit? Yung eye muscle area is highly correlated with more yield, mas maraming karne, and at the same time, highly correlated din sa growth rate. By combining two sources of information, mas mabilis din ang response pagdating sa growth rate. So we're working on that actually.
Okay po, thank you so much po, Doc Bing Flores. Now we have a questions again from Sir Joel Salazar from Pisau. And this question addressed to Dr. Atabay. Okay po, Doc Atabay, rephrase ko po yung question ko po kanina. So may we know the reproductive parameters for us to measure the reproductive efficiency of cattle and buffalo?
Reproductive parameters. So, unang-una pong ginagamit namin to measure efficiency after AI is the conception rate or the pregnancy rate. Kasi nadidetermine po namin yan dahil we have the mga ultrasound and other early pregnancy diagnosis tests. But then also, it can be by rectal palpation, ay malalaman din po natin yan, yung pregnancy rate at 60 days. And then after that is the calf production. So, which is really a measure of the fertility rate, yung being able to produce a calf from the AI. So, yun po yung mga important parameters na ginagamit namin. So, ang pinakamahalaga po talaga is the calf production.
So much again, Doc Bing Atabay. And yeah, we have another question po from Brian Bosito. And our question is for Dr. Flores. Doc Flores, hello po. Meron po tayong question to, this question is addressed to Dr. Atabay. Sabi niya po, good morning po, Dr. Atabay. What practical advances and innovations in livestock reproduction can we utilize for native animals?
For our native animals. So, yun nga po, we are continuously doing the upgrading. So, we are producing crucibreds. So, ganun din po yung ating mga ginagamit na mga biotechnology. Yung sa AI nga, and then, it's basically artificial insemination. Pero tinuturuan po natin sila na maging mga modifications, yung ating mga AI technician that we are conducting the seminar or training. So para may apply nga po natin tong matuto sila kasi wala namang ultrasound, yung palpation lang, manual palpation. But then yung dati kasi pag hormonal synchronization, basta hindi buntis, yun na yun, we are subjecting that to the program. But this time we are emphasizing yung just choose the animal with the structure, especially of the corpus luteum, kasi alam po nila yun na nakakapa naman yun. So, for higher efficiency. Kasi pag wala naman yung structure na yun, nagsasayang ka lang ng hormone. So, that also contributes to a lower efficiency. So, ngayon, yun po yung ina-emphasize namin. And then not only sa corpus luteum, but then also this time, ay ini-emphasize namin yung kung nakakapad na rin dyan ang follicle kasi kahit pag medyo may experience na po yung ating mga AI technician. So, yun po yung mga simpleng pagbabago na makakapataas po both in our whatever native and also in our dairy buffaloes. So, yan po. Of course, medyo ano po tayo ng hormone naman kasi hindi lang basta mag-induce tayo ng estrus. Lalo na po sa buffalo, napakalaki talaga ng problem sa reproduction compared sa cattle, no? So that's why the efficiency mas mababa sa kalabaw compared sa ano. So kailangan natin lalo na po yung nasabi nating summer infertility na madali silang maano sa heat stress. So kailangan po na tayo ng ovulation. I-induce natin din yung ovulation kasi not all animals that are in estrus will be able to ovulate. So ito po yung mga major advancement namin na ginagawa. And of course, may kaunting additional cost, but then at the end, it is more costly to maintain an unproductive animals than giving a little more, to the cost of the hormone or mga intervention. So, meron po kaming na-develop ngayon na kung gusto nyo ng super pangmayaman, so depende po sa efficiency natin. So na-develop na po namin yan through the years, and it's really addressing towards more efficiency in reproduction. Of course, hindi lang po sa buffalo, pati sa cattle.
Po, Doc Atabay. And now we have a new question from Sir John William Gamis. Hello, good day po. My question is for Doc Atabay. So can we apply or use FTAI, ovarian ultrasonography, and pregnancy diagnosis to small ruminants? If yes, is there any researches conducted that utilizes small ruminants using the mentioned technologies? Thank you po.
Oh, that's a very excellent question and query. So actually po, of course, we started the Epicsign AI and all this intensive reproductive management in buffalo. But recently, mayroon na po tayong ina-apply ngayon sa ating goat, or ito'y sa pangunguna po nila Dr. Lerma Ocampo, because they also have this project in reproduction in goats. So the fixed time AI now is applied in goat, which obviously increase their pregnancy and production, the production rate. So mayroon na po both ultrasound, fixed time AI, and in goat.
Thank you po again, Doc Atabay. And hello po, Doc Flores.
I'm here. Sorry.
Yeah.
Pala.
Opo.
Kalimutan ko na naman. Okay, yes, I'm here.
Opo. Meron na po tayong mga questions, ma'am, para po sa inyo. And ito po yung unang questions from Brian Bosito. May optimum or minimum number of microsatellite markers ba na required to form a reliable microsatellite panel for percentage verification?
Okay, so what we use is, we actually depende rin sa pinili mong markers, at saka depende kung how related the individuals are. When we started, sabi namin nun, 8 is okay. For cattle that are unrelated, actually, 8 is enough to give you enough of the confidence. But if animals are related, let's say may mga half-seeds ka dyan, you actually need to go more. So right now, ang workable namin, based on our experience with our current population, is we actually need to have about 15 to 16 microsat. Ang working set namin ngayon is 15 set of microsatellite markers to be able to eliminate yung mga possible non-parent and to be able to elucidate yung closely related individuals. So I hope that answers your question.
Okay, thank you so much, Doc Bing Flores. And last question po. Kung wala na pong tayong tanong. Okay. So, a question from Erica Lapus of UPLBFI. And this question is for Doc Bing Flores. So, sabi niya po dito, is the high-value breeds of carabaos already available for farmers? Or
Yes,
how long until those become available if not?
Actually
po,
yes, actually po, the genomically tested bull semen is already published in our online directory. So it's already available and it's being used already. Now, we do have what we call yung parang bull assignment per area. It's a matter of managing kasi yung anong klaseng mga genetics to avoid inbreeding in a particular area. But if you are actually really interested, you might want to get in contact with our coordinators at different centers. If that's your PLB, then we have a center in PCC, PLB, and sabihin niyo we are interested in acquiring the semen from the genetically tested bull. It's already available.
All right. So thank you so much po. Any question po from the webinar room? Okay. So kung may mga questions pa po tayo na hindi pa po natin na itatanong at bigla po nating maisip right after the presentation, o pwede po kayong mag-send sa amin through our Livestock Biotechnology Center Gmail, and susubukan po nating ipasagot ito sa ating mga experts, speakers po natin ngayon, kay Doc Atabay and Doc Flores, to address your questions po. Now, sa tingin ko po, medyo okay na po at nasagot na. So, yung ating mga katanungan ngayong araw na to. So, sabi nga po namin sa LBC, we couldn't have done this successfully without the expertise of our speakers who spared their time attending this webinar series. So, let us move on to awarding of certificate. So, let us hear what inside this certificate. Okay, so present this certificate of appreciation to Dr. Esther B. Flores for the time and expertise as a resource speaker during the Livestock Biotechnology Center Research Development and Extension Agenda Technical Caucus webinar series on September 3 to 4 to 2020, assigned Dr. Claro N. Mingala, Chief Livestock Biotechnology Center, and Dr. Yopresina P. Atabay, OIC Research and Development Division, Philippine Carabao Center. Thank you so much po, Doc Bing Flores, for your time po and expertise po. Maraming salamat po.
Okay, well appreciate.
Sa ulitin po, Doc. Thank you. Now, present the certificate of appreciation to Dr. Yopresina P. Atabay for the time and expertise as resource speaker during the Livestock Biotechnology Center Research Development and Extension Agenda Technical Caucus webinar series signed by our Chief Livestock Biotechnology Center, Claro and Mingala. Thank you so much po, Doc Bing Atabay, sa oras and sa expertise niyo po na naibigay po natin ngayon sa ating mga participants. Okay. And yeah, thank you everyone for participating this webinar session mula kahapon hanggang ngayon po. So we will be inviting you again for the last part of this webinar once we finalize the day through your email and text messages. So the presentations will be about animal nutrition, antimicrobial resistance, and disease diagnostic on livestock species. Now, we have come to the end of these sessions. Then I would like to invite all of us first for the photographic session, and please open your webcam. Okay, open na po yung ating webcam at nang makita po yung ating mga naggagandahan at naggagwapuhang. Yun. So, please po, paki-open. And habang nag-open pa po ng webcam yung ating ibang participants, so thank you so much po ulit sa ating mga participants from different government agencies and academic institutions. So maraming salamat po. Okay, so naka-open na po yung ating webcam. Okay, and 1, 2, 3. Okay, once again, 1, 2, 3. Okay, thank you so much. And once you leave today's web assessment form, and we would appreciate if you would complete that and provide your feedback. On behalf of, thank you again for joining us today. And we will see you again for the last part of this webinar series. And on behalf of the Livestock Biotechnology Center and PCC, I'm Louis Baligad, signing off.